Samantha Jensen, who's going to be talking to us about the human side, the host side of things. So we can sequence Cocci, we can sequence humans, and at some point we'll be able to merge those two, hopefully, and get some real insight into the host pathogen interaction. So take it away, Sam.
All right. Thank you so much for this opportunity. I'm really excited to share some of our results that we are finally able to have into common variants that may contribute to risk for disseminated coccidioidomycosis. So as you're all well aware, there is a broad spectrum of severity in symptoms between different patients. I'm going to focus today on the differences between patients who have pulmonary-only disease and the very small number of patients that have disseminated disease. For the purposes of this talk, I've grouped the wide variety of patients into three categories. So the first is uncomplicated valley fever. So that's anyone who's asymptomatic or has just a very uncomplicated disease restricted to the lungs. And then complicated pulmonary coccidioidomycosis, which is individuals who have some kind of complication, but it's still restricted to the lungs. So cavitation, having a chronic disease that's not resolving quickly, or even respiratory failure would fall into that category. And then finally, disseminated coccidioidomycosis, which is any kind of dissemination to the skin, to any organ, bone, and then of course, coccidioidal meningitis. So the basic idea of host susceptibility to coccidioidomycosis is that we know that there are environmental factors. We know that, if you're working with dirt all the time, you're much more likely to get disease. We know that if you are male, you're more likely to have disseminated coccidioidomycosis. And we know that there are inborn errors in immunity, like Dr. Su has published on, that can influence whether or not you get disseminated disease. One of the most controversial has been that many of you have shown in your epidemiological studies that individuals who have certain races or ethnicities are significantly more likely to have dissemination. So this is a meta-analysis we did of 6 very good papers over the last, I think 50 years. And you can see here that individuals who identify as white or Caucasian are significantly less likely to have dissemination than those of any other race or ethnicity group. And then I'd like to quickly point out that individuals who are Black or African American or Filipino also have significantly higher risk of dissemination. But where does this race or ethnicity factor fall between environment and genetics? Now, it's almost entirely likely that it's both. But since I'm a geneticist, I'm interested in which part of this risk is genetic. So in order to answer that question, we've collected a cohort of 635 patients. 361 of them have uncomplicated disease, 90 have complicated pulmonary disease, and the other 184 have disseminated disease. We have very similar results when we make the same odds ratio chart with our data. You can see here that among the 83 individuals who identified as white or Caucasian, they did have a significantly reduced risk of dissemination relative to anyone who'd identified as something else. And then we were also able to replicate the results in individuals who considered themselves Black or African American and individuals who considered themselves Pacific Islander or Filipino. I would note, though, that we only had 10 individuals who considered themselves Pacific Islander or Filipino, and so that's why that odds ratio confidence interval is absolutely massive. Now, that's still not telling us anything about whether or not it's genetic, right? That's still just psi, or self-identified race or ethnicity. And so in order to move beyond that to the DNA level, we calculated what's called genetic ancestry. And I talked about this before, so I'll try and be quick. So the first method we use to calculate genetic ancestry is what's known as principal components analysis. A lot of you use that. Basically, it's a dimension reduction algorithm. And so you put in all of the variants in the genome, and then you pull out components that represent variability amongst the group. So this plot right here is showing the locations where we got our several thousand reference samples from. We didn't actually collect them ourselves. These are in a database. But you can see that there are individuals, there are locations where patients, and samples were collected in Africa, in the Americas, in Europe, and then in South Asia and East Asia. And when you look at the principal components plot, this is PCs 2 and 3, you can see that those continental ancestry is somewhat clumped together. And I've drawn a green circle around the references that have African ancestry because when we plot our samples, so these are now our samples, and the colors represent which sire these individuals said that they were, you can see that individuals who said that they were Black or African American fall within this circle pretty well. So they match what we see in the genetic ancestry of our reference samples. And then if we color this time by disease severity, you can see that there are a lot more orange dots which represent disseminated disease within this circle than there are in the others. This is just another way of representing that matches what I showed you previously with the sire. So again, people who have primarily European genetic ancestry by this method are significantly less likely to have dissemination. And then people who have primarily African ancestry by this method are significantly more likely to have dissemination. So the second method we use to quantify genetic ancestry is what's called admixture. It gives you an estimate of the proportion of variants from a genome that are from a given reference population. So in this case, each one of these columns represents an individual, one of our patients. And you can see here, so this person has more than 50% of their genome that's estimated to come from East Asian ancestry. And then the remaining portion is primarily from European ancestry. Whereas the person next to them, even though they also identified as Pacific Islander or Filipino, has primarily actually admixed American ancestry. So maybe some kind of indigenous group that's more similar to the Americas than to these East Asian references. And when we segregate again by severity, we see that individuals within this green, which is also African genetic ancestry here, are significantly more likely to have disseminated disease than they are to have uncomplicated disease. So individuals with high proportions of African ancestry are much more common in our disseminated group than they are in our uncomplicated group. So the final piece that you need to know to understand the GWAS results is local ancestry. So everything I've showed you so far has been measures of global ancestry, which is across the entire genome, what's the primary ancestry that we see. Whereas local ancestry is at this specific position, due to crossing over or recombination, at this specific position, what are the alleles? What's the likely ancestry they come from? So in this toy example, it would be population 1, which is that dark purple, and then population 3, which is the lime green. Okay, so we finally got to the GWAS methods. We used these 6 different methods that are implemented by a kit called Admix Kit, which uses either global or local ancestry to correct for these differences in populations so that we can use all of our samples at the same time to search for common variants. And again, I don't have time to go into all of them. I'm just going to go into one result. So I'd love to talk to you after this about more of the results. It'll just take longer to step through. So here are the 10 different loci that we found. You can see every method that we used found at least one locus. And the one that I'm going to focus on for purposes of the talk, since I have not that much time left, is this chromosome 1 hit, which is found. Okay, so this is a Manhattan plot. So the y-axis represents the negative log 10 value of the p-value of the association between having that genotype and having disseminated disease. So the higher the number, the more that variant is associated with dissemination, either positively or negatively. So this is our hit here in chromosome 1. You can see it falls within this non-coding RNA, but importantly, it's upstream of these 2 immune genes. So this is interferon alpha inducible 44 and 44L. They are paralogs of each other, and they have both been associated with risk for infectious disease, including COVID-19, Staphylococcus aureus, hepatitis C, a bunch of other things. So that's very interesting to me. When you look at just the allele frequencies between the 3 groups, UVS, CPC, and DCM, you see that you can see a difference. You can see the increase in DCM, but there's not significant. If I do a statistical test, this is not a significant difference. And that's because Tractor, which is the method that found this association, corrects for both global and local ancestry. So if we break it down by global ancestry, I'm just showing you European ancestry, you can see that this association is a lot stronger. We've got a significant p-value of the difference in allele frequency between individuals who have dissemination and who have uncomplicated disease in this European group. And then if you look at the local ancestry, so now we're looking at the number of alleles. So there are 126 alleles that came from disseminated patients that also had European local ancestry. Then you see an even stronger association between allele frequency and whether or not someone has dissemination. Very confusingly, and that's why it's taken me so long to get through all of this GWAS data, the effect sizes are not the same in different populations. And that's because there's differences in linkage disequilibrium and differences in allele frequency between populations. And so if we look in just our patients that had primarily East Asian global ancestry, which just isn't that many patients, but those individuals who had disseminated disease, none of them have the alternate allele for this variant. And so in our patients who have East Asian local or global ancestry, the effect size is actually reversed. So if you have this variant, you're much less likely to have disseminated disease in these East Asian populations. And if you're European, you're more likely to have disseminated disease. So there's a lot of really cool things to get into here with all of the rest of our things. But moving on, we wanted to know how exactly are these GWAS variants affecting the phenotype in the end? So one possible thing we can explore is, are they affecting the expression of different genes? And so just briefly, you can see here at the top of the chart, this variant I've been talking about in chromosome 1 is actually a known EQTL expression quantitative trait locus for this interferon-inducible 44. And it's associated with an increase in expression of that gene. So then we did a genome-wide differential expression analysis. So this is in a subset of our patients, 246 that had RNA-seq, and that number is increasing every day. But for now, it's 246. And you can see that there's a lot of differentially expressed genes. You can see CLEC4D here, which is it's a paralog of CLEC7A. And you can also see interferon-inducible 44 and 44L. When we do a gene ontology enrichment analysis for these samples, we see what we expect, which is a lot of immune-related phenotypes, including regulation of cytokine production. So in conclusion, we found that genetic ancestry is associated with risk for dissemination. We found that using ancestry-aware methods, we were able to identify 10 new loci associated with risk for dissemination. And we found that there are immune-related differentially expressed genes in our patients in DCM, including that interferon-inducible 44 and 44L. So I hope that once the manuscript is available, you'll all read it and tell me what's wrong and what's good. And I hope that as we continue doing this analysis, including rare variants analysis, that we'll be able to help many more patients. So thank you to everyone at UCLA, UC Davis, and the Valley Fever Institute, as well as to our funders, including the U19 grant, and to the nurses and patients. And I'll take your questions if we have any time. So thank you.
Thank you so much. It's been really fantastic to see the evolution of this project. I think many of us have followed this for the last few years, and this update is just fantastic. So with that, any follow-up questions from folks? So you said you have a lot of other, well, okay.
Fantastic.
Thank you. Oh, that's a great question. My favorite kind. Yeah.
Rebecca.
That was a great presentation.
Thank you.
I'm trying to wrap my medical, clinical, and public health brain around how to translate that to individual and population health.
Yes.
And I would love just a little bit of a linkage. So potentially, could there be a patient who is diagnosed with Cocci, and then we somehow look at their genome, which I feel like we could do a lot quicker now,
Yep.
and then predict their risk of dissemination? Is that kind of where this could be going?
Yeah, so this is most relevant for your patients that don't have a known immune deficiency.
Right.
So this is like, why are we getting people who are healthy that all of a sudden have disseminated disease? Where does that come from? And so, yeah, we could do something like a polygenic risk score eventually, probably not with this level of data, although I could make one, it would just be not very predictive. But yeah, eventually we would be able to have your patient do their sequencing, say, what's your risk of developing disseminated disease, et cetera. And maybe you could use that like way in the future for like deciding whether or not you give antifungals to someone who has only pulmonary disease for now, right? Something like that. That's not anywhere close to happening, but that's the idea.
Gotcha. And that's the tricky part is if they have the risk, what do we do then? So
Exactly.
awesome. But this is a great building block. Thank you.
Yeah, I'm excited. Thank you.
One more question.
So my thought is that you're going to be born and you're going to get a genome. And then that's going to make all kinds of predictions about which drugs you should take and what dose it should be and what risk you have for X, Y, and Z. And that would be the way we practice medicine.
It's the advent of personalized medicine, right? So this is
Scary
where we're going.
potentially helpful.
Hopefully helpful.
Yeah.
Go ahead, sorry.
I guess I was wondering if you could talk a little bit about that one factor that you talked about that like really peaked. What the sort of what the reverse is, like what is the frequency of that among these groups that we see that have higher rates of dissemination, and then sort of like I know that your talk is focused on the genetic factors, but sort of what you see as as a geneticist, what you see as the role of the environment, because I think we also know that there are a ton of other environmental insults that really vary across these different racial and ethnic groups. And I think we don't know a ton about like long-term insults to the immune system. And that structural environmental factors that
you're absolutely right.
these
Like there's
disparities.
huge epigenetic differences between racial groups because of weathering, which is essentially if you're in a group that's constantly being starved or under stress, you're going to have differences in how your genes are expressed regardless of what the actual genotype is. This is only testing the genotype. So there's genetic things that can be affected by environment. There's environmental things like diet, the location you live. Those are all affected by the environment. And honestly, I'm a geneticist, but I believe that those probably have a stronger effect on whether or not you get the disease and whether or not you get disseminated disease than the genetic factors, the common genetic factors. That's my guess. I can't remember what the other question was.
Just like what the reverses of that like factor that you identified, like is that in fact meaningfully higher like in frequency? Like if you were like among this population, what is the
Okay. So
percentage that
the
have
variant is most common in East Asian populations where it's sometimes the major allele, so more than 50%. And it's least common in European ancestry populations where it's around 10% normally. In our data set, it's much higher and lower for the East Asian. So
But
that's
you found that it was sort of like quote unquote protective in East Asia.
in East Asians
but
protective.
the opposite.
Yep.
Okay. I'll talk to you later.
Yeah,
But
no,
anyway,
I'd love
that
to
was
talk
really
to you. Yeah,
interesting.
anyone who wants to talk through, I can show you the other variants. It'd be great for me. So
Yeah,
thank you.
Thank you so much. Yeah,